Preparation model paper 29. I'm N. Shankar Reddy, PhD plant pathology. First one is sugarcane ratoon stunting is actually in continuation to the previous videos. Sugarcane ratoon stunting disease is caused by here I have mentioned old name. Old name in the sense this sugarcane ratoon stunting is generally now it is known to be caused by Lifesonia xylem subspecies xylem. So previously it is called as Clavibacter xylem subspecies xylem. Now it is called as Lifesonia xylem subspecies xylem, and there is a reason to ask this because in 2019 probably maybe 2021 not 19 2021 this question was asked like giving sugarcane ratoon stunting is caused by but in the option they have never given this Lifesonia xylem subspecies xylem, but Clavibacter xylem subspecies xylem option was there. So the old name is Clavibacter xylem subspecies xylem. Now it is called as Lifesonia xylem subspecies xylem. Banana moko wilt disease is caused by which race of Ralstonia? So generally this moko wilt is caused by race 2. There are some races are there, right? If you see race 1, race 2, race 3, race 4, race 5. Race 1 infects Solanaceae, race 2 Musaceae or banana, race 3 potato, race 4 ginger, race 5 mulberry. So like that there are 5 races are there in this Ralstonia. I repeat once again, race 1 Solanaceae, race 2 Musaceae or banana, race 3 potato only, race 4 ginger, race 5 mulberry. So that is that is the generally races that they the crops that they infect. Apple proliferation is caused by, so apple proliferation is actually a phytoplasmal disease which is caused by a phytoplasma. The next one is first plant pathogenic bacterium to be completely genome sequenced. They're asking a pathogenic bacterium, plant pathogenic, sorry. So if it is a normal bacterium to be completely genome sequenced, it is Haemophilus influenza. Since they have mentioned the plant pathogenic bacterium, that is Pierce disease of grapevine, Xylella fastidiosa. Right, first plant pathogenic fungi to be completely genome sequenced is rice blast, Magnoporthe, Greciae or Pyricularia varese. So in 23 probably 21 probably, yeah, they have asked what is the genome sequencing related to bacteria. They have mentioned first human bacteria that is completely genome sequenced, and plant pathogenic bacteria like Haemophilus influenza and Xylella fastidiosa they have given in a in a same option only. So to have the knowledge regarding the 2021 paper, we have already published a 2021 net paper. So please go to the playlist section and see the net question papers, previous net question papers, where we have started from 2006, 7, 9, 14, 14, 1, 14, 2, 18, 18, 1, 18, 2, 2019, 21, 23, up to 23 we have published. 21, you please go through that section and see this question was asked. So genome sequencing related to bacteria, something like that. First plant pathogenic bacteria and fungi I have told. So to have a better understanding, on the next slide I have placed the genome sequencing of the plant pathogenic organisms. Generally the next one is downy mildew genera can be classified based on, so downy mildew genera like plasmopora, sclerospora, peronospora, bremia, all the downy mildew genera are generally classified based on the sporangiopore character. For example, if you see the downy mildew, generally the symptoms can be observed on the lower surface, so the sporangiospores are hanging in the reverse direction like this, right? So this sporangiopore characters, based on how the sporangiospore are branchings, are based on the sporangiopore characters and branching, it is classified. So generally sporangiospore characters, sometimes maybe sporangiospore characters and branching also will be there in the options, but general answer is the sporangiospore characters. The 207 bud eye symptoms is produced by, so in if you see there is only one anthracnose, especially in grapes anthracnose, which produces a bud eye kind of symptoms like this. This is the grape, which produces exactly this kind of symptom, that is known as bird eye symptoms, that especially we can observe on grapes anthracnose, which is caused by Elisino amphelin, I think. The next one is a citrus greening indicator. So indicator plant for citrus greening. So generally indicator plant for citrus greening is a sweet orange. So the indicator plants that we will place in the next slide. So there are different kinds of indicator plants in the plant virology. So that I will place in the next slide. So generally citrus greening indicator plant is sweet orange. The indicator plant for potato spindle tuber viroid. So for potato spindle tuber viroid, generally tomato can be used as the indicator plant for potato spindle tuber viroid. The next one, tufts of flagella at both ends can be observed in which type of uh, that is what type of bacteria, I mean type of flagella they are asking. So if the tufts of flagella are present on the both side, it is called as lopotrichus. So lopotrichus kind of flagella having a tufts of flagella at both sides, that is known as a lopotrichus. So for our understandings, again I will place the different types of flagella in bacteria as well. So this is genome sequences of all plant pathogenic organisms. For example, especially plant pathogenic organisms. If you see the first fungi to be completely genome sequenced, that is a plant pathogenic fungi, Magnoporthe greciae, the pathogen which causes disease called rice blast. So that was first sequenced by D. Natal in the year 2005. So for our additional information, I have mentioned the genome size as 40 MB as well. So first bacteria genome sequencer is Xylella fastidiosa. If it is a human bacteria, Haemophilus influenzae, AJG Simpson 2000. 2.68 MB. First DNA virus completely genome sequencer is cauliflower mosaic virus, sequenced by Frank et al. 1980. First RNA virus to be completely genome sequencer is tobacco mosaic virus, sequenced by Golet et al. 1982. So generally the size is 6.3 to 6.5 kbp kilobase pairs. The next one is viroid. First viroid to be completely genome sequenced is potato spindle tuber viroid, that was sequenced by Grass 1978, Dixon 1977, 79. Generally 359 nucleotides this potato spindle tuber contains. So the yeast is also the first generally fungi to be completely genome sequenced. So generally if they are not mentioning the first fungi to be completely genome sequenced, then it goes to Saccharomyces. If they are mentioning first plant pathogenic fungi, it is a Magnoporthe greciae, that is a Pyricularia variae. Maybe based on the options also it may varies. So just take a look into this. So that was sequenced in the year 1996. Size is 12.1. Actually this yeast was sequenced by a group of organization. The nematode, first nematode to be completely genome sequenced is Chinorhabdis elegans, that was sequenced by Andrew Fire and Craig Mello in 1998. Size is 100 MB. Red bread mold Neurospora crassa, which was sequenced by Gallagher et al. 2003. The size is 40 MB. So these are all the general some organisms first sequences and their sequenced persons and their information as well. Hope this will be useful because in the previous years, in the last 4-5 years, there might be 1 or 2 questions from this. And when it comes to only plant pathogenic bacteria, only bacteria to be completely genome sequenced, these are all the couple of bacteria that is a completely genome sequence. So Xylella fastidiosa 2.68, that was sequenced by A.J.G. Simpson year 2000. Ralston solanacearum 5.81 MB, it was sequenced by C.A. Broucher 2001. Xanthomonas axinopodis 5.26, Xanthomonas campestris, Pathovar campestris 5.07, which was sequenced by Anasier da Silva 2002. Xanthomonas variae, Pathovar variae, that is the rice bacterial leaf blight 4.94 BM Lee. 2005 it was sequenced by Pseudomonas syringae, Pathovar tomato 6.54 MB. The size is genome sizes AR Colomer, that was sequenced in the year 2003. Xanthomonas variae, Pathovar variae cola, that was sequenced by Salzburg. I was a little bit confused about their genome size and the year of sequencing. Please let us know if you guys know about this genome size and sequencing year of the Xanthomonas variae, Pathovar variae. So this is about genome sequencing, especially related to bacteria. Coming to the next one, these are all the indicator plants in the previous slide that I have told in the slide one, right? So the virus indicator plant is a goose foot, which is a Chenopodium amaranth. Citrus greening indicator plant is sweet orange. Citrus sinensis. Citrus exocortis is indicator plant is trifoliate orange, which is citrus trifoliate. Citrus tristeza virus indicator plant is Claxi lime, we can also call as Mexican lime. Scientific name is Citrus aurantifolia. Product spindle tuber viride, tomato, Solanum lycopersicum, Solanum berthollet, and red rot of tea, which is the indicator plant is fish poison bee. And the last one is a phytoplasma. The indicator plant is a periwinkle. So these are all the couple of indicator plants and the pathogenic organisms used for detection. And the types of bacterium, the types of flagella, right? A trichus means A means no, trichus means hair-like structure, that is generally we can call as a flagella, right? A trichus, which means no flagella are present. Example is xylella. This is a trichus example. Mono, the name itself indicates mono means single. Trichus means hair or flagella-like structure. A single flagellum is present at one end of the bacteria, and only one side. Example, xanthomonas. You can see only single flagellum at one side of the bacteria. Amphitrichus, a single flagellum in both ends of the bacterium. You can see here Amphitrichus, a single flagellum at both sides of the bacterium. Best example is Pseudomonas species. Peritrichus, peri means tufts of flagella or large number of flagella all over the body. If you see here, this is peritrichus flagella, contains a multiple number of flagella all over the body. This is called peritrichus kind of flagella. We can observe in Erwinia, Pantovia, Escherichia coli, and Clostridium. Lopotrichus, so a tufts of flagella at both sides. This is, we can say, example. So multiple number of flagella is present at both sides. A tufts of flagella. Best example is Spirulina. Cephalotrichus, a group of flagella at one end. We can see here in cephalotrichus. Cephalo means head. So for head, hair is present at only one side, right? So like that you can remember. So cephalo means generally head-like structure. So for a head, we have a hair at only one direction. So in cephalotrichus kind of flagella, tufts of flagella or multiple number of flagella are present at only one side. Best example is Pseudomonas fluorescens. Circumtracheal or subpolar flagella, which means lateral flagellum at one side or both side may be present. So the best example is Agrobacterium. And in continuation, in plains of India, Phytophthora infestans survives as generally in the infected tubers. This Phytophthora survives in the plains. Generally, if you see in mostly in cool areas or cool climates, only this potato grows well. In such kind of area, this Phytophthora survives inside this potato tubers. So generally they have asked in plains of India. So in plains of India, in any plains, generally this Phytophthora infestans can survive in the infected tubers. The next one is the tetrazolium chloride is used for the identification of which pathogen. So there are some kinds of detection tests for the identification of the pathogen. Generally, this tetrazolium chloride is used for the identification of banana bunch-top virus. I think on the next slide we might have different kinds of identification tests for the various kinds of plant pathogens. We'll see there in detail. But generally, this tetrazolium chloride is used for BBTV, banana bunch-top virus. This banana bunch-top virus is transmitted by an insect vector called black aphid or aphid, Pentalonia nigronervosa. The next one is groundnut bud necrosis. Groundnut bud necrosis is transmitted by Thrips tabaci. First plant pathogen recognized in the world. So generally it is actually a parasite. Loranthus, we used to call it as Loranthus or Dendroptoi or Mistletoe or giant mistletoe. These are all the various kinds of name. The first recognized plant parasite, first recognized plant pathogen in the world is Loranthus. So Loranthus is the best example for partial stem parasite. Complete stem parasite is a Cuscuta. Partial stem parasite, Loranthus. So Loranthus is known to be the first recognized plant parasite or first recognized plant pathogen in the world. This Loranthus is also known as the Dendroptoi or Mistletoe or giant mistletoe, which was first recognized by Albertus Magnus in the 1200 AD. This Albertus Magnus is generally probably his church priest, I think. Single ascus Cleistothecium with dichotomously branched appendages can be observed in which kind of powdery mildew? So to have a general understanding about this, the kinds of powdery mildew, again I have given a detailed description on this next slide. Anyway, the answer is a single ascus. They are asking single ascus. First we need to understand whether it is a single ascus or multiple ascus. Okay. It is clear it is a single ascus. The next one is the Cleistothecium is a dichotomously branched. So dichotomously branched appendages with single ascus is the characteristic feature of Podospora. Anyway, don't worry about this, that I will tell you in the next slide. So single ascus with dichotomously branched is a characteristic feature of Podospora. The next one is Hortic nets are found. It is very important and repeatedly asked question from the couple of previous nets. Generally, Hortic nets are found in ectomycorrhiza. Ecto means outside. In endomycorrhiza, Hortic nets are absent. Stem gall of coriander is caused by Protomyces macrosporus. Peach leaf curl is caused by Taphina deformans. Agrocin K84 is a biocontrol agent. BCA means biocontrol agent. Recommended for which disease? Generally, this Agrocin 84, Agrocin K1026, which are sold outside with the trade names of Digal and Nogal, are the biocontrol agents recommended for Agrobacterium tumefaciens, that is the crown gall of stone fruits or apple. So this crown gall of stone fruits, which is caused by Agrobacterium tumefaciens, this Agrobacterium tumefaciens management, the best recommended practice is biocontrol agent, the two Agrobacterium K84, Agrobacterium K1026. Their name, general name is Agrobacterium radiobacter. We can also call it as a Rhizobium radiobacter. Okay, this Rhizobium radiobacter or Agrobacterium radiobacter, which are two strains are available, K84,026. Those are recommended for this management of Agrobacterium tumefaciens. Pore size of laminar airflow chamber 0.3 micron or micrometer. So fungicide which is applied prior to fungal infection, which is applying prior, so before infection, it is called protectant fungicide. If you are applying something prior, it is called protectant kind of fungicide. And this slide represents to the previous slide, that is actually the identification test we have discussed, right? So this tetrazolium chloride test is recommended for banana bunch-top virus identification, right? So there are other identification tests are here for the detection of various kinds of plant pathogens here. Generally, NaOH test or sodium hydroxide test generally recommended for this kernel bunt of wheat and ergot diseases also. Brain flotation technique is recommended for wheat tundu disease. And ooze out test, we know very well all bacterial diseases we can recommend ooze out test. For viruses, ELISA can be recommended, enzyme linked immunosorbent assay, which was first given by procedure was given by Adam and Clark 1977. And about ELISA in the previous lectures also we have discussed many times. In upcoming lectures also in detail about this ELISA will be there. We may expect at least one or two questions from this ELISA as well. Tetrazolium chloride test is recommended for the detection of BBTV, banana bunch-top virus, which is transmitted by black aphid pentaludium nigronervosa, that I already discussed, right? So iodine test, RTV, rice tumor virus. Page or STS page, polyacrylamide gel electrophoresis. Sodium dodecyl sulfate polyacrylamide gel electrophoresis. Fluoroglucinol test. So these are all the various identification tests that can be recommended for the identification of viroids. So DAPI technique. So generally DAPI technique is used for identification of phytoplasma. And paraquat technique is recommended for post-harvest disease. These are all the various kinds of identification techniques and the pathogens used for the identification. And regarding the types of appendages, right? See here I have divided into two, one ascus containing and many ascus. So hope you guys know what is ascus and what is ascospore. Ascus is a sac-like structure, or you can also remember as a bag-like structure, not a big deal. Okay, ascus is a sac-like structure where 8 number of ascospores are packed inside. So ascus means which are present inside. sorry, ascospore which are present inside a sac-like structure is known as ascus. This overall structure is called ascus, and inside ascospores are present. So based on the number of ascus, one ascus or many ascus, we have divided one ascus and many ascus. These are all the clistothecial appendages. Okay. So one ascus, so myceliaid appendages in one ascus, example is Parotheca. This is a one ascus myceliaid appendages. Sparotheca. Dichotomously branched appendages. Sparotheca. We can see here dichotomously branches. So branches are dichotomous, right? So dichotomously branched appendages in one ascus is Podospera. They have asked in the previous one is dichotomously branched with one ascus is a Podospera. Okay. Now we will go to the section of many ascus. So in the many ascus, coiled, circinoid, or maybe coiled at the end, or curved at the end, various kinds of terminologies they use. We can see here this kind of appendages. If you see, this is a uncinella, coiled appendages or circinoid appendages. We can see uncinella. Bulbous appendages with many ascus, phylactinia. Bulbous appendages we can see at the base of the clistothecium. We can see bulb-like appendages, right? So this is the best example for phylactinia. Myceliaid appendages in many ascus. If it is a single ascus, it is Parotheca. Many ascus means Ericife, powder mildew genera, and Levilula. This is the myceliaid appendages. And next one, dichotomously branched. Single ascus means Podospera. Multiple ascus means, or many ascus means Microspera. We can see dichotomously branched with many ascus. Microspera. So these are all about various kinds of appendages or types of appendages on the clistothecium. The next one is food storage organ of bacteria. Generally, bacterial food can be stored in an organ called polybeta hydroxybutyrate. We can also call it as hydroxybutyrate. This question was asked in 23. Give an example of both apoplast and symplast fungicide. Versatile aluminum and metaloxyl are the best examples. Agar-agar is made from, agar-agar is made or produced from red algae or sea algae. There are two species of red algae or sea algae can be used. There are hundreds of species might be there, but there are two species that we can hear vigorously. One is the Gelidium species. The next one is the Gracilaria species. Gelidium species and Gracilaria species are the red algae, or we can also call it as the red sea algae. That is used for the production of agar-agar. So agar-agar is a solidifying agent, right? So oleander leaf scorch is caused by Xylella fastidiosa. Like you can see, there are various kinds of disease that is caused by this Xylella. I will tell you the list. Pierce disease of grapevine, almond leaf scorch, oleander leaf scorch, alfalfa disease, phony peach leaf curl. So these are all the various kinds of disease that is caused by Xylella fastidiosa, and it is the first plant pathogenic bacterium also to be completely genome sequenced. So this oleander leaf scorch is caused by Xylella fastidiosa. Virides consist of nucleic acids. So virides generally consist of a nucleic acid, single stranded RNA. If I want to tell you, positive single stranded RNA. So there are so far 40 virides have been discovered, reported. All the 40 virides contains positive single stranded RNA as a genetic material. Tobacco necrosis satellite virus was first reported by Casanis. So Casanis was the first person who reported this tobacco necrosis satellite virus. Shape of a bacterial growth curve. So they are asking growth curve. So generally this bacterial growth curve is sigmoid in sigmoid curve. Black heart of potato is a oxygen deficiency. Generally black heart of potato is due to oxygen deficiency. That is very important. White tip nematode. Generally white tip nematode of rice is Aphelingoides basi. Root knot nematode is Maladogyne. Irvinia belonging to the family Enterobacteriaceae. So Irvinia belonging to the family Enterobacteriaceae. Here I have given since the food storage organ of bacteria in the first one 221 polybeta hydroxybutyrate, right? And again I have given a couple of other food storage stainings, staining organs and all those things. Endospore staining organ is malachite green, plasmid staining organ argandide, capsule staining organ ink. So the question is food storage organ, that is a polybeta hydroxybutyrate. So how to identify this? We must stain to identify whether it is present or not, right? So these are all a couple of staining agents which is used for the identification of the requirements. So for the generally endospore identification, we recommend malachite cream. For plasmid staining, we recommend acridine dye, capsule Indian ink, flagella staining, carbol fission, lichen staining agent, archin, inclusion body staining agent, azure, and cytoplasm and nucleus staining agent, acetic acid, acetic dye, bacterial food storage organ, that is poly beta hydroxybutyrate, that is Sudan black B. So these are all the different kinds of staining agents. In demicyclic rust, which stage is absent? It is a very important one. Lack of iridial stage is known as demicyclic. Lack of iridial stage is known as demicyclic. In a demicyclic rust, which stage is absent? Iridial stage. So such kind of questions was asked. This was asked in 2021. Lack of iridial stage is known as demicyclic. So SAR, systemic acquired resistance, is induced by the external application of. So systemic acquired resistance we can activate by the application of salicylic acid. So by the application of jasmonic acid and ethylene we can activate ISR, induced systemic resistance. Founder of mycoplasmal bacterial research. So Kleine-Berger Nobel, generally considered as founder of mycoplasmal research. So she discovered L bacteria, L form of bacteria. So why she named L form of bacteria? She was working in a Lister Institute. To honor the Joseph Lister, he is also a very big famous microbiologist. She named as L form of bacteria that was discovered by Kleine-Berger Nobel in 1935. Inverted T-shaped foot cells, phylides, vesicles are found in the which fungus? So inverted T-shaped foot cell is observed in generally observed in Aspergillus. So the main difference between Aspergillus and Penicillium is inverted T-shaped foot cell. So inverted T-shaped foot cell is observed in Aspergillus, whereas it is absent in Penicillium. The next one is which fungicide is considered as a cosmetic fungicide. There is a fungicide called tridamorph, which is considered as a cosmetic fungicide. So diencisteins produces the identification color. So this diencistein are fluoroglucinol, diencistein and one more stain which is there, which is used for the identification of pytoplasmal diseases. Just now we discussed in the previous slide, right? So these two tests are used for the identification of pytoplasma, which produces a color called blue color. PCR was developed by Carey Mullis, who invented the PCR in 1983, which is a polymerase chain reaction, which is used for the multiplication of DNA. So the enzyme used in PCR, there is a special enzyme called Taq polymerase, Thermus aquaticus. The enzyme name is Taq polymerase, which is produced from a bacteria called Thermus aquaticus, which is a heat-resistant bacteria. So that can withstand up to 90 to 95 degrees Celsius. Expand PCR, polymerase chain reaction, that is the expansion of PCR. 241, denaturation temperature of PCR. Generally, this in PCR there are three steps are there. Denaturation, annealing, and extension. So the temperature may varies from PCR to PCR. For example, if you see some PCRs may denaturate the DNA at the rate of 92 to 94. Some PCRs may denaturate at 95. So it may varies. Generally, the denaturation temperature is considered to be as 92 to 94 degrees Celsius. Denaturation. The next process is annealing. Annealing can happen at 55 to 65. And the last one is extension. So extending the DNA, that happens at 72 degrees Celsius. So this is very important. So every PCR have their own denaturation and annealing temperature. But if you see extension, almost all PCRs having the standard extension temperature, that is 72 degrees Celsius for the extension of DNA. The next one is the candidatus pytoplasma accursinus sieve phloem concept. So this candidate pytoplasma only occurs in sieve tubes. This concept was actually given by Doe et al. Because that's why he discovered this candidate as pytoplasma, that is Mulberry Doer first pytoplasmal disease in 1967, right? So he also gave one more reason. This phytoplasm is only present in phloem sieve tubes, mainly because of presence of excess amount of sterols. Because this phytoplasma requires excess amount of sterols for their growth. This phytoplasma and spiroplasma can be only managed by antibiotics, especially penicillin is highly ineffective. Tetracycline is highly recommended for the management of the cytoplasmal and spiroplasmal diseases. Muriform septations in conidia can be observed in. So muriform type of septations or conidia can be mainly observed in alternaria. We can see bell-shaped conidia like this muriform, alternate vertical and alternate form of septations, like both vertical and horizontal kind of septations can be observed in this conidia, muriform conidia. And bacterial blight of P, Pseudomonas syringae, patho or PC can survive in. So the Pseudomonas syringae, generally most of the bacterial disease can survive in seeds, majority of the plant bacterial diseases. So some may be spread through, like wind splash, rain splash, overhead irrigation through. So such kind of things might be there. L form of bacteria means cell wall lacking bacteria. Just now we discussed, right? L form of bacteria, cell wall lacking bacteria, that was first discovered by Kleiniberger Nobel in 1935. To honor the institute, she named it as the L form of bacteria, which means cell wall lacking bacteria. The first cloned plant toxin gene is isolated from. So the first toxin, indirectly they are asking first toxin isolated from. So the first toxin is isolated from wildfire of tobacco disease, that is caused by Pseudomonas syringae, patho or syringae. Okay, so that is the first plant toxin gene to be isolated. The next one is gram-positive bacteria contains the amount of mucopeptides. So gram-positive bacteria contains around 85% of mucopeptides. Gram-negative contains 3 to 12% of mucopeptides. Gram-negative bacteria lack of which acid? So gram-positive bacteria contains teichoic acid. Gram-negative bacteria lacks of teichoic acid. Which pathogen enters into the plant cells respiration? So let me go once again. When a pathogen enters into the plant cells, respiration increases due to. So for example, imagine if one plant pathogen is entering into the plant cells, the plant cells respiration generally increases due to binding and unbinding of oxidative phosphorylation. So due to this process, the respiration is automatically increases in the plant cells. So budding spores are generally known as blastospores. Blastospores are known as budding spores. Endospore forming bacterial genus. There are two important genus which form this endospores, Bacillus and Clostridium. Legislative method of plant disease control is known as a quarantine. We can also call as exclusion. Sorry. Legislative method, right? That is exclusion. Okay, it's not quarantine is the overall method, right? And the both answers will come in generally. If in the option only quarantine will be given, if only a quarantine was there, go with the quarantine. If there is no quarantine, exclusion was there, then go with the exclusion. So this answer is the legislative method of plant disease control is both quarantine and exclusion will come. Citrus gummosis is caused by Phytophthora species. Phytophthora, Phytophthora, various kinds of Phytophthora are responsible. There are, it has been reported that there are 6 species of Phytophthora responsible. Phytophthora, Phytophthora palmivora, Phytophthora syringae, something like that. There are 6 species are reported. Generally, Phytophthora species is responsible for the citrus gummosis. Phytophthora is also one species responsible. And in autoclave sterilization, 15 psi 30 minutes at what temperature? So the autoclave temperature is 121.6 degrees Celsius. If sometimes options 121.6 was not might be there, so then go with 121 degrees Celsius, not a big deal. 121.6 degrees Celsius or 121 degrees Celsius for 15 psi for 15 to 30 minutes. Cucumber mosaic virus is transmitted by. So cucumber mosaic virus generally transmitted by sap transmission and also transmitted by aphids. So if it transmitted by 2 means it is called as bimodal transmission. Best example, cauliflower mosaic virus also can transmit by 2 aphids and as well as sap. And example of haploid, I repeat, haploid sexual spores. If you see, ascospores and basidospores are the best example of haploid sexual spores, whereas oospores and zygospores are the best examples of diploid sexual spores. Again, in the sexual spores, ascospores and basidospores are considered as non-resting spores, whereas oospores and zygospores are considered as resting spores. The next one is extracellular PGPR. Bradorhizobium is generally considered as extracellular PGPR. Maybe other things also might be there. Prunus necrotic ring spot virus is transmitted by, it's a very important. This Prunus necrotic ring spot virus is transmitted by pollen. ICTV started. International Committee on Taxonomy of Virus was started in the year 1973. So before this, ICTV was called as ICNV. ICNV means International Committee on Nomenclature of Virus, which was actually established in the year 1966. The same ICNV renamed to ICTV in the year of 1973. ICTV means International Committee on Taxonomy of Virus, which was renamed from ICNV in 1973. Blossom end rot of tomato is caused by, blossom end rot of is actually a calcium deficiency. Leaf wetness period of apple scab, so it is considered as a 9 hours. Minimum leaf wetness period of 9 hours is required for occurrence of apple scab. How to remember this apple scab 9 hours? In apple 5 letters are there, in scab 4 letters are there, 5 letters for 4 letters, 9 letters, so consider like a 9 hours, like that. Okay, apple scab contains 9 letters, so that 9 hours, like that you can remember. Okay, enzyme involved in the viroid replication, there is an enzyme called RNA polymerase 2. So for virus replication, RNA polymerase 1 is useful, but when it comes to viroid replication, RNA polymerase 2 is used. Durable cell wall can be seen in which family of bacteria? So generally gram-positive bacteria, that is a firmicutes, contains a durable kind of cell wall, right? The next one is vivotoxin example. Best example is fusioric acid. So the fusioric acid is the best example for this vivotoxin. The term vivotoxin was given by Diamond and Wagner in 1953, and the concept was given by T.S. Sadasivan. Father of soil solarization, Jacob Cotton is generally considered as father of soil solarization, sorry, and he's the only father still alive, in the sense like in all the father related to all the other fields are almost died, but there is only one father as far of my knowledge, soil solarization father only is still alive, Jacob Cotton. Please let me know if there is other fathers are alive, other fathers in the sense fathers related to the particular discoveries or concepts. Okay, but father of soil solarization is Jacob Cotton. The next one is parasexuality was discovered by. Parasexuality was what is parasexuality first of all? So generally in sexual reproduction of fungi, plasmogamy, karyogamy, meiosis generally occurs in a sequence. So here in parasexuality, it never occurs in a sequence, or sometimes not at specified points. It occurs at unspecified points. So this concept was first discovered by Pontecorvo and Roper in 1952 in a fungus called Aspergillus nodulans. So this is also important. Which fungus parasexuality was discovered? Aspergillus nodulans. Like that we can expect. The term phytoalexin was introduced by Muller and Borger in 1940. They introduced the term phytoalexin, and also they are the one who discovered the world's first phytoalexin, that is a pisatin. Okay, which is isolated from pea plant. Kittelsen killer invented in the year. This question was asked in 2021, 1952. Kittelsen killer is otherwise called as Captain. Why it is called Kittelsen killer? Because it was invented by Kittelsen. That's why it is known as Kittelsen killer. This Captain general recommended for seed treatment and other purposes as well. But it was invented in the year 1952. Soda borax. So burgundy mixture is also known as soda borax. It is a very important one. Again in 2020 when this question was asked. Soda borax is otherwise called as burgundy mixture, is otherwise called as soda borax. We can also call it as Mason lime because it was developed by Mason in 1887. That's why it is known as Mason lime or soda borax or burgundy mixture. The next one is environmental factors in relation to plant disease was first given by. So the involvement or the importance of environmental factors in disease development, spread, all those things was first given by J.E. van der Planck. He is considered as father of plant disease epidemiology. Nucleic acid 271. Nucleic acid is the infectious material of plant virus. This question was repeatedly asked in the previous nets. I saw this question for 3 times, around 2 times I think. Nucleic acid is the infectious material of plant virus was first given by. So before it was thought that even W.M. Stanley thought that protein coat is the infectious material, but not. Nucleic acid is the infectious material which is causing the infection. This discovery or this was first given by Geirer and Scram in 1956. Here is also important. Nucleic acid is the infectious material of plant virus was first given by Geirer and Scram 1956. Piliform ascospore is produced. Piliform ascospore is generally produced by claviceps. So claviceps produces this kind of ascospore that is called generally piliform ascospore. Okay, Podospora leucotricha produces how many ascus? They produce only one ascus. Ascus is a sac-like structure where ascospores are packed inside. So this is a Podospora leucotricha which causes what powdery mildew? Rose powdery mildew, I think, right? Podospora leucotricha, which produces only one ascus. Size of common bean mosaic virus 12 into 750. This is a little bit important. Size of bean common mosaic 12 into 750 nanometer. Potato leaf curl virus genome. So this potato leaf curl contains a genome of single-stranded DNA. Simple interest diseases produces a saturation curve. Compound interest disease produces a sigmoid curve or S-shaped curve. Okay. Polyetic diseases produce bimodal curve. So there are three concepts are there in epidemiology. You guys know, right? Simple interest diseases, compound interest diseases, and polycyclic disease. Simple interest diseases are known as monocyclic. Compound interest diseases are known as polycyclic. Polyetic disease generally. So generally the simple interest diseases or monocyclic disease produce a curve called saturation curve. Polycyclic or compound interest disease produce a curve called sigmoid curve or S-shaped curve. Polyetic is bimodal curve. Turmeric leaf blotch is caused by. So turmeric leaf blotch is caused by a fungi Taphrina maculans. Banana streak virus is transmitted by leaf hopper. Just a minute. Banana streak. Okay, anyway, banana streak is transmitted by mealybug, not leaf hopper. Banana streak virus and cocoa solen soot virus. This two and other things might be there. Banana streak and cocoa solen soot virus are transmitted by mealybug. And little leaf of brinjal is transmitted by leaf hopper. Monospicitis. ICNV established in the year ICNV, International Committee on Nomenclature of Virus, 1966. That was renamed as ICNV, International Committee on Taxonomy of Virus, in 73. Mycoparasitism is in between. So generally myco means fungi. Mycoparasitism is between fungi and fungi. Hypersensitive response is also known. Hypersensitive response observed in which crop? So generally the hypersensitive response was first time observed in a crop, tobacco. 8th edition of Berg's Manual of Determinative Bacterial was edited by. So it was edited by Buchan and Gibbons. So this question was not asked, but in many books this question was there. 8th edition of Berg's Manual of Determinative Bacterial was edited by Buchan and Gibbons. And the next one is Protomyces macrosporus, which causes coriander stem gall. Protomyces macrosporus. The matured conidia can spread through. Generally, this matured conidia can spread through air. That is, we can also call as wind, right? Windborne or airborne. So bendy yellow vein bendy yellow vein virus is transmitted by. So this bendy yellow vein virus is transmitted by white flies. Both systemic and contact fungicide example. You can take an example of phosphatyl aluminum or metalaxyl also. Which bacteria produces a blue color stain while adding to the milk? There is a bacteria called Pseudomonas syncytia or syncasia which produces blue color stain when you are adding to the milk. And there are a couple of bacteria which produces blue, red. For example, you can take red Serratia marsans, blue, this Pseudomonas syncytia. Some kind of bacteria will be there. In detail that we have discussed already in the bacteriology section. PCR can amplify both the DNA and RNA. I was a little bit doubted about it because some people are telling only DNA can amplify it, and some people RNA also can be amplified by using PCR. But in majority of the literature it is mentioned that PCR can amplify both DNA and RNA. So please let me know if there is any corrections regarding this PCR amplification. And secondary fixing agent in electron microscope, there is an agent called osmium tetroxide that can be recommended as a secondary fixing agent in electron microscope. Pseudomonas syringae producing a toxin is called as Pseudomonas syringae produces a toxin called a TAB toxin. We can also known as wildfire toxin, which is produced by Pseudomonas syringae pathovirus syringae. Which secretion system is found in almost all plant pathogenic bacteria? Almost all plant pathogenic bacteria contains a type 1 kind of secretion system. There are almost 7 kinds of secretion system are there, right? Type 1, type 2, type 3, type 4, type 5. Type 4 agrobacterium twin fashion. Type 3 is the most dangerous one, pathogenicity, and like that we used to study, right? But almost all plant pathogenic bacteria contains type 1 kind of secretion system. Regarding the secretion system, we have mentioned in detail about in the bacteriology section. Role of inoculum development in Phytophthora, rate of inoculum development in Phytophthora infestans. Generally, this Phytophthora infestans comes under polycyclic disease, which develop their inoculum at the rate of 0.3 to 0.5 units per day. I repeat, 0.3 to 0.5. Dispersal of pathogen through humans is known as anthropochory. If the disease spread through humans, it is known as anthropochory. IPS means Indian Phytopathological Society. IPS was started in the year of 1948, started by B.B. Munkur along with some other famous people like S.R. Bose under the chairmanship of S.R. Bose. B.B. Munkur actually started this in 1947 February 28, and it came into enforcement and action in 1948. The trade name of COC means copper oxychloride. It is actually selling with the trade name of blue copper or Blitox 50 outside. Monograph of Synchytrium was written by Synchytrium endobioticum, which causes potato wart. So this Synchytrium monograph was written by J.S. Carling in 1958 probably. Year is not confirmed, but it was written by J.S. Carling. Wheat stem rust repeating spore. So iridospore is considered as a repeating spore. Teleospore is considered as a resting spore. Sexual stages of rust fungi was discovered by Craig. So Craig in 1927 he discovered the sexual stages are zero stages, are pycnial stages, are spermatial stages. So the zero stage, sexual stage, or spermatial stage, or stage 0, it was discovered by Craig 1927. Best example of autoecious demicyclic rust. So the autoecious demicyclic is rubus orange rust, white pine blister rust. So these are the examples of autoecious demicyclic. Demicyclic means lack of iridial stage. So autoecious demicyclic example is rubus orange rust and white pine blister rust. So here just now we have discussed regarding the question number 297 and 298. We can see here rust iridospore. Iridospore is also known as the repeating spore, also known as summer spore. The condition is N+N, the color is golden brown. With generally single celled. For the management of rust, we can recommend oxycarboxyn, the trade name is Plantvax. Smut is generally teleospore, which is resting spores or winter spores. Condition is N+N, black or dark brown in color, which is actually bicelled. Best example is teleospore. For the management of smuts, we can recommend carboxyn, the trade name is Vitavax. Rust generally localized infection, very less on flowers, maybe probably 1%. We can see on foliar parts also, but flowers very less. Smut generally systemic infection, mostly we can see inflorescence and as well as grains. So these are all the various kinds of monographs. I'm just going fast. Pythium monograph, G. Rangasamy, Alternaria Varma, Helminthosporium AP Mistra. Monograph of downy mildew and powdery mildew, DM Spencer, Cercospora RS Vasudeva. In India, in world level, it was written by Chep. Fungal spore Cercospora, again Chep. Taphrina Agemix, Erysipelas Ubran. These are all the various kinds of, I'm just going through fast. And Phytophthora Irwin and Ribeiro, 71 probably or 77 probably, I'm not sure. Fusarium C. Booth, it's a very important. Monograph of Fusarium was written by C. Booth, very important. Helminthosporium AP Verma, Smut Kalman Venki. And other monographs have been given here. Please go through yourself. Since one monograph was seen earlier, so I have mentioned couple of monographs and who wrote and all those things. You guys please go through it. And for further more information, students can refer my book, A Vision into Plant Pathology, a complete student version. For further doubts and clarification, students can reach us at www.geekresearcher.com. Stay geeky and stay tuned. We are Team Geek Researchers.